Welcome to Biomania Revision Podcast.
It's great to be back. And today we're doing something a little different, aren't we?
Completely different. Usually we'll pick up some articles, some source material, and just see where the conversation takes us. But today, we're on a mission. We're doing a dedicated deep dive revision session, and our target Chapter 11, Photosynthesis in Higher Plants.
A classic. It's really the bedrock of plant physiology. It's pretty tough to understand the rest of biology without getting this down first.
I agree. And for everyone listening, whether you're a student cramming for an exam or you're just curious about how the world works, this session is for you. We're going to break down the core material, concept by concept. But before we get into the nitty-gritty of molecules and diagrams, I want to start with a really essential question.
Go for it.
How do plants actually eat? I mean, we call them autotrophs. We say they make their own food. But what is the machinery inside that actually does that? It just seems like magic sunlight hits a leaf and poof, you get sugar.
It does look like magic from the outside, but it's actually the most sophisticated physicochemical process on Earth. And why is it so important? Because at the end of the day, pretty much every living thing depends on it.
Because it makes food, right?
That's one of two reasons, actually. The first, yes, is that it's the primary source of all food on Earth. Even if you eat meat, that animal ate a plant. But the second, and equally important reason, is that this process is responsible for releasing oxygen into the atmosphere. Without the machinery in this chapter, there'd be no oxygen to breathe.
So the stakes for this chapter are pretty high.
Absolutely. It's a matter of life and death, literally.
Okay, so here's our roadmap for today's session. We're going to start out like detectives. We'll look at the early history to see how we even figured all this out. Then we'll zoom into the chloroplast to look at the pigments. After that, we'll get into the machinery, the light reaction, how energy is captured, and the dark reaction, how the sugar is made. And finally, we'll look at the factors that control the speed of this whole factory.
That's a logical flow, from history to structure, then the process, and finally, regulation.
Exactly. So let's time travel. Section 1. The detectives. We're heading back to the 1700s. Who's our first character?
Joseph Priestley. The year is 1770. Now, you have to remember, back then they didn't really know about oxygen or carbon dioxide in the modern sense. Priestley performed a very famous series of experiments with a bell jar.
I love the visual of this one. You just put a glass jar over a burning candle.
Right. And what happens if you put a jar over a burning candle?
Well, it'll burn for a little bit, and then it'll just go out, suffocate.
Exactly. Priestley observed that a burning candle somehow damaged the air. He noticed the same thing with a mouse. If you put a mouse in a sealed bell jar, it would eventually suffocate. The air became foul.
A bit of a grim experiment, but the point is clear.
But then came the twist. Priestley placed a mint plant inside that bell jar, along with the mouse or the candle.
And what happened?
The mouse stayed alive, the candle kept burning.
So the plant was doing something to the air?
Priestley's conclusion was that plants restore to the air whatever breathing animals and burning candles remove. He didn't call it oxygen back then, but he understood that plants were fixing the air.
Okay, so plants are air purifiers.
Yeah.
But then along came Jan Ingenhousz. He took Priestley's setup and tweaked it a bit, right?
Yes. Ingenhousz asked a critical question. Does this happen all the time? Does it happen at night? He set up a similar experiment, but used an aquatic plant. And he noticed something very specific. When the plant was in bright sunlight, tiny bubbles formed around the green parts.
Bubbles.
Yes, bubbles. But in the dark, no bubbles. None at all.
So this restoring of the air only happens when the lights are on.
Precisely. He established that sunlight is essential for this process, and it was later identified that those bubbles were, in fact, oxygen. So Ingenhousz proved that it's only the green parts of plants that release oxygen, and only in the presence of light.
Okay, so we've got air restoration and a light requirement. Now let's jump forward to 1854. Julius von Sachs. This sounds like the Industrial Revolution of biology.
In a way, it was. Sachs provided the evidence that plants are actually factories producing glucose. He showed that when plants grow, glucose is made, and it's usually stored in the form of starch.
And starch is just a long chain of glucose, isn't it?
It is. And Sachs also pointed out that this green substance, which we now call chlorophyll, isn't just floating around anywhere in the cell. It's located inside special bodies.
Meaning the chloroplasts.
Correct. So Sachs gave us the factory concept. The green parts are the factory floor, the product is glucose, and the storage inventory is starch.
I get it. Now, the next experiment is my favorite because it's so visual. T.W. Engelmann.
Ah, the prism experiment. This was brilliant. Engelmann took a prism and split light into its spectral components, the rainbow, VHUR. He then shone this rainbow onto a green alga, Cladophora.
So he's just put a rainbow on some algae. Sounds peaceful.
But he added a twist. He placed the algae in a suspension of aerobic bacteria.
Aerobic, meaning bacteria that need oxygen to survive.
Exactly. These bacteria were like little oxygen-seeking missiles. Wherever photosynthesis was happening, oxygen would be released, and the bacteria would flock there.
So where did the bacteria gather?
They accumulated mainly in the blue and red light regions of the spectrum.
That's interesting. Not in the green region?
Nope, not in the green, mostly blue and red. This is the first time anyone had described an action spectrum for photosynthesis. And it turned out that this pattern closely mimics the absorption spectra of chlorophyll A and B.
We'll come back to those pigments in a moment. But there's one more detective left. Cornelius van Niel. I hear he completely changed the equation.
He was a microbiologist. He wasn't looking at plants, he was studying purple and green bacteria.
And these bacteria don't use water, right?
That's the key. These bacteria use hydrogen sulfide H2S instead of water. And when they photosynthesize, they don't release oxygen, they release sulfur.
Wait, let me stop you there. So if they use H2S and release sulfur, does that mean that in plants, which use H2O, the oxygen must come from the H2O, not from the CO2?
Brilliant. That's exactly the inference Van Niel made. Before him, many people assumed the oxygen came from the carbon dioxide, CO2. But Van Niel demonstrated that water is the hydrogen donor. The water molecule is split, and oxygen is a byproduct of that split, not of the CO2.
That's a massive shift in understanding. So the equation becomes CO2 plus water in the presence of light yields a carbohydrate plus water plus oxygen.
Correct. And this was later definitively proven using radioisotopic techniques.
Okay, so the detectives have cracked the case. We know light splits water to release oxygen and helps make sugar. Now let's go to the crime scene. The factory floor. Section 2. The site and the pigments.
We're going inside the leaf, specifically into the mesophyll cells. These cells are packed with chloroplasts.
And the source text mentions something interesting about their alignment, doesn't it?
It does. Chloroplasts are dynamic. They actually align themselves along the walls of the mesophyll cells. They'll orient themselves so their flat surface is parallel to the wall to catch the optimal amount of light.
Like tiny solar panels tracking the sun.
Exactly like that. Now, if we slice a chloroplast open, there's a clear division of labor inside. It's a two-part system. You have the membrane system and the stroma.
The membrane system includes the grana, right?
Yes, the grana and the stromal lamellae. This is the light division. Its job is to trap light energy and to synthesize ATP and NADPH.
Yeah, the stroma.
The stroma is that fluid-filled matrix. That's the dark division, or the enzymatic division. This is where the sugar is actually synthesized.
I want to clarify something here because it always confuses students. When we say dark reaction, it does not mean it happens at night.
That is a very common misconception. It doesn't mean it happens in the dark. It just means it's not directly driven by light. However, it is entirely dependent on the products of the light reaction, the ATP and NADPH. So if the light goes out, the dark reaction will stop a short time later because it runs out of fuel.
Got it. So dark reaction is just a name, not a timestamp.
Correct. We prefer to call it the biosynthetic phase.
Much better. Now let's talk about colors. If I look at a leaf, it looks green. But if I do paper chromatography, separate the chemicals, I don't just see green.
That's right. A chromatogram will show you 4 distinct pigments. You'll see chlorophyll A, which is a bright or blue-green. Then chlorophyll B, which is yellow-green. Then you have the xanthophylls, which are yellow, and the carotenoids, which are yellow to yellow-orange.
Why so many? Why not just have one super efficient green pigment?
It's all about efficiency. If you look at the graph in figure 11.3, chlorophyll is clearly the chief pigment. Its absorption peaks in the blue and red regions match the rate of photosynthesis almost perfectly.
So chlorophyll A is the boss.
It's the reaction center. But the other pigments, THLB, xanthophylls, carotenoids, we call them accessory pigments, they act like an antenna. They absorb wavelengths of light that chlorophyll might miss and then transfer that energy over to Kirale.
So they're casting a wider net, catching more light.
Exactly. And they have another crucial role, protection. They protect chlorophyll from photooxidation. They act as a shield so the main engine doesn't get burned out by high-intensity light.
Smart. Okay, we have the factory, chloroplast, and the workers. Pigments. Time to turn the machine. Section 3. The light reaction.
also known as the photochemical phase. This is where the action starts. We're talking about light absorption, water splitting, oxygen release, and the formation of our high-energy intermediates, ATP and NADPH.
The source material talks about photosystems, PSI and PS2. And I remember the numbering is a bit confusing.
Yes, the numbering is based on the order of discovery, not the order of function. In the actual process, photosystem 2, PS2, comes first.
Okay, so PS2 is the starter.
It is. Each photosystem is a complex. It consists of a light harvesting complex, LHC, which contains hundreds of those pigment molecules we just discussed, the antenna. And then there's a single reaction center, which is a specific chlorophyll, a molecule.
And they are sensitive to different wavelengths.
There's a slight difference. In PSI, the reaction center peaks at 700 nanometers, so we call it P700. In PS2, it peaks at 680 nanometers, so it's P680.
Okay, now explain the Z scheme to us. This is the electron transport, right?
Yes. Think of it like a relay race. We start at PS2. Light at 680 nanometer hits the antenna pigments. That energy gets funneled to the reaction center, P680. An electron in that chlorophyll molecule gets excited and jumps out of its orbit.
It escapes the atom.
It does. It gets picked up by an electron acceptor. Now, this electron is in a high-energy state. It gets passed downhill through an electron transport system, which includes cytochromes.
Downhill meaning it's losing energy.
In terms of redox potential, yes.
Yeah.
And as it moves down, that released energy is used to make ATP. But the electron's journey isn't over. It arrives at photosystem I.
But light is also hitting PSI, isn't it?
Exactly. Light at 700 nanometer hits PSI. Its own electron gets excited and jumps out to another acceptor. The electron coming from PS2 simply fills the empty spot, the hole left behind in PSI.
Okay, so PSI fed PSI. But where did the electron from PSI go?
It moves downhill again, but this time it's passed to a molecule of NADP plus AC. It reduces it, forming NADPH plus H plus AC.
So we made ATP during the transfer, and we made NADPH the very end. But wait, if PS2 keeps losing its electrons, isn't it going to run out?
That is the million-dollar question. PS2 needs a constant supply of replacement electrons. This is where the splitting of water comes in.
The engine room.
Specifically on the inner side of the thylakoid membrane. An enzyme complex splits water. Two molecules of H2O are broken down into 4 protons, H+, one molecule of oxygen, O2, and 4 electrons.
And those electrons go to PS2.
Yes, they replace the ones that were lost. And the oxygen, it's released into the air. That's the very oxygen we breathe.
It's amazing to think that every breath we take began with a water molecule being split inside a thylakoid membrane.
It connects us directly to this microscopic process.
Now, you mentioned making ATP. This brings us to section 4.
Yeah.
Photophosphorylation.
Correct. And there are 2 types. The process we just described, where electrons go from PS2 to PSI and then on to NADPH, is called non-cyclic photophosphorylation because the electrons don't come back.
And it produces both ATP and NADPH.
Right. But there's another mode, cyclic photophosphorylation.
When does that happen?
This occurs when only PSI is functional. It typically happens in the stromal lamellae
Why there?
because the membranes of the stromal lamellae lack PS2 and they also lack the NADP reductase enzyme. So the excited electron that leaves PSI has nowhere to go but back. It cycles back to PSI through the electron transport chain.
And does this make NADPH?
No. No NADPH, no oxygen, it only synthesizes ATP. This likely happens when light wavelengths are beyond 680 millimeters, which can't excite PS2.
So cyclic is just an ATP generator. Non-cyclic is the full package.
That's a great way to put it.
Now we have to get a little technical. The chemiosmotic hypothesis. This explains how ATP is actually made. It's not magic, it's mechanics.
It's a game of gradients. Think about a hydroelectric dam. To generate electricity, you need high water pressure on one side and low on the other. In the chloroplast, we want to create a high concentration of protons, H+, inside the thylakoid lumen and a low concentration in the stroma outside.
A proton dam. How do we build up that gradient?
In 3 ways. First, remember the water splitting? That happens on the inner side of the membrane, so the protons from water are dumped directly into the lumen.
Step 1, fill the reservoir.
Second, as the electrons are moving between the photosystems, a carrier molecule actually picks up a proton from the stroma on the outside and pumps it into the lumen on the inside.
Step 2, pump in even more.
And third, on the stroma side, the NADP reductase enzyme consumes protons from the stroma to make NADPH, so it's actively removing protons from the outside.
So we're filling the inside and emptying the outside. The difference in concentration and therefore pH must be huge.
It's massive. And this proton gradient contains potential energy. To break the gradient, the protons want to rush back out into the stroma, but they can only go through one specific gate.
ATP synthase.
Correct. ATP synthase has two parts, CF0, which is embedded in the membrane forming a channel, and CF1, which protrudes out into the stroma. The protons flow with great force through the CF0 channel. This flow causes the head of the enzyme, the CF1 part, to rotate.
That's a literal turbine.
It is. And the energy from that rotation is used to catalyze the formation of ATP from ADP and an inorganic phosphate.
That is incredible. A molecular turbine powered by a proton dam that was created by sunlight.
Nature's engineering is pretty hard to beat.
Okay, so now we have our bags of energy, ATP and NADPH. But we still haven't made any food. We've just charged the batteries. Section 5, the biosynthetic phase, the dark reaction.
This is where we finally fix CO2. This process happens in the stroma.
And we know about this largely thanks to Melvin Calvin.
Yes. After World War II, Calvin used radioactive carbon-14 to trace the path of carbon in algal photosynthesis. He discovered that the first stable product formed was a 3-carbon acid PGA, or 3-phosphoglyceric acid.
Which is why we call it the C3 pathway.
Exactly. But here's where a fun little mystery popped up. Scientists figured that if the product is a 3-carbon molecule, then the acceptor molecule that grabs the CO2 must be a 2-carbon molecule. 2 + 1 = 3, simple math.
Seems logical.
But biology loves surprises. They spent years searching for this 2-carbon compound and found nothing. Eventually, they discovered the acceptor was actually a 5-carbon ketose sugar ribulose bisphosphate, or RoBP.
Wait, 5 + 1 is 6?
Yes, but that 6-carbon intermediate is highly unstable, and it immediately splits in half to form 2 molecules of the 3-carbon PGA.
Ah, very sneaky. Okay, so let's walk through the 3 stages of the Calvin cycle.
Stage 1. Carboxylation. This is the most crucial step. CO2 combines with RoBP. The enzyme that does this is called RuBP carboxylase oxygenase, Rubisco, the most abundant enzyme on Earth. This reaction creates the 2 molecules of 3-PGA.
Stage 2.
Reduction. This is where we spend our energy. For each CO2 molecule fixed, this series of reactions uses 2 ATP and 2 NADPH on the path to forming glucose.
And stage 3.
Regeneration. If we don't remake the RuBP acceptor, the whole cycle will grind to a halt. This step costs 1 ATP.
So let's do the math. To make a single glucose molecule, which has 6 carbons, how many times does the cycle have to turn?
1 turn fixes 1 CO2, so to get 6 carbons, you need 6 turns of the cycle.
And the total cost?
18 ATP and 12 NADPH.
That is an expensive molecule.
It is, but glucose is a high-energy molecule. You have to spend energy to make energy.
Fair enough. So this is the standard C3 pathway. Most plants do this. But then we have some rebels. Section 6, the C4 pathway.
The Hatch and Slack pathway. This is an adaptation found in plants from dry tropical regions. Think of maize or sorghum.
What's the problem with C3 in the tropics?
It's the heat and water stress. But mainly, C4 plants have evolved a way to avoid a wasteful process called photorespiration, which we'll talk about in a moment.
How is their structure different?
They have something called Kranz anatomy. Kranz is German for wreath.
A wreath of cells.
Exactly. Around their vascular bundles, they have a ring of large cells called bundle sheath cells. These cells have thick walls that are impervious to gas, they have no intercellular spaces, and they are packed with chloroplasts.
And the process, the first product, is different.
Yes. In C4 plants, the first stable product is a 4-carbon acid oxaloacetic acid, or OAA.
Walk me through the steps. This sounds like some kind of smuggling operation.
It's basically a CO2 pump. Step 1 happens in the mesophyll cells. Here, the primary acceptor is not RoBP. It's a 3-carbon molecule called PP, or phosphenolpyruvate.
And the enzyme.
Pepcase. This is critical. The mesophyll cells of C4 plants lack the Rubisco enzyme. They only have pepcase. Pepcase grabs CO2 and forms the 4-carbon OAA.
What happens then?
This C4 acid, which can be malic or aspartic acid, is transported into the bundle sheath cells. Once inside, it's broken down. That's decarboxylation releasing CO2.
So they smuggled CO2 into this inner chamber and then released it.
Precisely. Now, inside the bundle sheath, the concentration of CO2 gets really, really high, and Rubisco is waiting there. Because the CO2 is so concentrated, the Calvin cycle can run very efficiently.
And the leftover 3-carbon molecule goes back to the mesophyll to become PEP again.
Completing the cycle.
So C4 plants essentially spend extra energy to pump CO2 to a place where a Rubisco can work without distractions.
Correct. It's like a turbocharger for an engine. It allows them to maintain high productivity, even in high temperatures and intense light.
You use the word distraction for Rubisco. That brings us neatly to Section 7, photorespiration, the wasteful process.
This is Rubisco's design flaw, if you want to call it that. Look at the full name. Ruby P carboxylase oxygenase. It can bind to CO2, but it can also bind to oxygen.
It's not very loyal.
It's competitive. If CO2 levels are high, it binds CO2. But if oxygen levels are high and CO2 is low, which happens in hot weather when plants close their stomata to save water, then Rubisco starts binding with oxygen instead.
What happens then?
Instead of making useful precursors for sugar, the reaction produces one molecule of phosphoglycerate and one molecule of phosphoglycolate.
Does that make sugar?
No.
Does it make ATP?
No. In fact, the plant has to spend ATP to clean up the mess, and it actually releases CO2 in the process. It's the opposite of photosynthesis.
That sounds like a disaster.
It's a net loss for the plant. But C4 plants get around this because their CO2 pump ensures that the concentration around Rubisco in the bundle sheath is always high, so Rubisco never really gets a chance to see the oxygen.
That's why maize can grow so fast in the summer heat.
Exactly.
Okay, finally, let's touch on the factors that affect this whole process. Blackman's law of limiting factors.
Blackman's law from 1905 basically states that if a process depends on several different factors, its rate is determined by the factor that is nearest to its minimal value.
The weakest link theory.
That's it. You could have all the light in the world, but if the temperature is freezing, the enzymes won't work. Temperature becomes the limiting factor.
Or if you have perfect conditions but no CO2.
Then CO2 will limit the rate.
What about light? Can you have too much light?
You can. At low light intensity, the rate increases linearly, but saturation is reached at just 10% of full sunlight.
Only 10%. Does that mean for most of the day, plants are getting more than enough light?
In an open field, yes. Light is rarely a limiting factor in nature, unless you're in a dense forest or underwater. In fact, very high light can be damaging. It can cause the breakdown of chlorophyll, which is called photooxidation.
Plants can get a sunburn.
Effectively, yes. We call it solarization.
Wow. We have covered the entire journey. From a ray of sunlight hitting a leaf, to the thylakoid, splitting water, pumping protons, making ATP, fixing CO2 in the Calvin cycle, and even the C4 workaround.
It really is a remarkable system.
So, to wrap up this revision deep dive, we've learned that photosynthesis is the engine of life. It's a precise, elegant, molecular dance.
And it leaves us with a provocative thought. Rubisco is arguably the most important and abundant enzyme on the planet, so why is it so inefficient? Why does it make this mistake of binding oxygen, leading to photorespiration? Is it just an evolutionary relic from a time when Earth's atmosphere had very little oxygen?
That's a fascinating question. Maybe evolution just hasn't fixed it yet, or maybe the C4 pathway is the fix.
Perhaps.
Well, that wraps up our revision of Chapter 11. Say like and tell how it was, and if you want more chapters to revise like this, and tell in comment section.
Happy study.